Welcome to the Deep Dive. We're jumping into something truly fascinating today, brought to us by you, this idea that brain function isn't just about the chemical signals we always hear about or the electrical zaps,
Right.
but that it also involves physical and mechanical forces. You shared some really intriguing material looking into how actual mechanical strain in brain tissue might interact with its chemistry and electricity.
Yeah, absolutely. And this deep dive is really centered around unpacking a pretty, well, maybe radical concept from that material you sent over. It's proposing this incredibly complex and crucially bidirectional relationship between the mechanical strain, the physical forces within the brain structure, its electrical activity, and the whole chemical landscape, the neurotransmitters.
So we're exploring how this whole system might organize itself and what that tells us about big things like sleep, maybe even dreams.
Exactly. We're hoping to uncover how this dynamic system works and maybe, what it means for those fundamental states.
Yeah. Okay, so get ready for some moments where things might just click into place. Because looking at the brain this way, mechanics, chemistry, electricity, all tangled up, it really does offer some surprising insights. Okay, let's unpack this.
So when we usually think about how the brain works, right, the focus is nearly always on neurotransmitters.
Yeah, dopamine, serotonin, GABA, all those chemical messengers. That's what you learn in biology class.
Exactly, how they bind to receptors, drive thoughts, moods, everything. And that's obviously super important, no question.
But this perspective, the material sometimes calls it brain kinematics. It suggests something extra.
That's the term, yeah. It suggests that neural strain flow, basically, the tiny movements and deformations within the brain's physical structure itself might be just as fundamental in shaping the brain's overall state.
Okay, strain flow, like physical movement shaping brain states.
And the core idea, the real kicker here, is that it's not just a one-way street. It's a bidirectional feedback loop, meaning the physical strain affects the chemistry, and the chemistry, along with the electrical activity, affects the physical strain back again. It's a constant conversation.
Okay, let's break that down. Start with strain affecting neurotransmitters. The material mentions some specific ways this could happen.
It did. One really key link they propose involves something called mechanosensitive ion channels.
Mechanosensitive. So sensitive to mechanics, to physical force.
Precisely. Think of them as tiny gates in the neuron's membrane that literally open or close when they're stretched or squeezed. Channels like TRP or POP or piezo-1 and piezo-2 are examples.
So if the brain tissue around a neuron is under strain, these gates physically get pushed or pulled open.
You got it. And when these specific channels open, they often let calcium ions flow into the neuron.
And calcium is the trigger for releasing neurotransmitters, right?
Exactly. It's the signal that tells the neuron, okay, release those chemical messages at the synapse. So physical strain in one spot could directly crank up or dial down neurotransmitter release right there.
Wow. And it's not just the neurons doing this. What about glial cells like astrocytes?
Good point. They're also sensitive to strain. And that's really important because astrocytes are workhorses when it comes to managing neurotransmitters, especially glutamate and GABA, the main excitatory and inhibitory ones.
So strain on astrocytes could change the local balance between go and stop signals.
Potentially, yes. Their response to physical force can alter how they handle those neurotransmitters.
Plus, there's the blood flow connection, neurovascular coupling.
Right, that's the mechanism where more brain activity means more blood flow to that area.
Right.
Now, if this model holds, and high activity often means high strain,
then strain could indirectly call for more blood.
yeah, signaling for more fuel, more raw materials, essentially the precursors needed to actually make more neurotransmitters. The physical demand could be driving the supply chain.
Okay, I'm following. Strain influences chemistry. Now, flip it. How do the chemicals and the electricity influence the physical strain flow?
Well, the electrical fields generated by all those ions moving around the very currents carrying the signals, they don't exist in a vacuum. They interact with the physical environment.
How so?
These tiny electrical forces can subtly modulate or tweak the neural strain flow. They can influence those phase waves, the rhythmic electrical patterns we measure with EEG.
Can you give an example? Like, how would a specific neurotransmitter do that?
Okay, think about acetylcholine, ACH. The material suggests that high levels of AC can boost theta wave activity. And interestingly, theta waves seem linked to more rhythmic cyclic patterns of strain.
Ah, and AC is dominant in REM sleep, isn't it? That's a neat connection.
It is. And then consider GABA, the main inhibitory transmitter.
The calming one.
Right. When GABA is dominant, overall firing goes down. And this seems linked to what the material calls strain desorption. It's like the physical tension in the tissue is actually releasing, dissipating, kind of a physical relaxation, mirroring the chemical calming.
And dopamine.
Dopamine seems tied into regulating more focused, maybe directed strain flows needed for specific cognitive tasks. So it's linking the physical dynamics to actually thinking and processing.
This leads us to another concept from the material that sounded pretty complex: stationary stress-straining waves.
Yeah, it sounds complicated, but the idea is maybe simpler than the name. Think of them not as waves traveling across the brain,
Okay.
but as stable, resonant patterns of tension and relaxation that kind of exist within specific regions.
Yeah.
Like, a standing wave on a guitar string. It vibrates, but stays in one place.
Ah, okay. A stable pattern of physical stress locked in place.
Exactly. A stable pattern where the physical strain and the local electrical rhythm, the phase waves, are locked together, resonating.
And why does that matter? What's the point of these stable patterns?
The idea is that they create these stable physical microenvironments, and these environments act like consistent chemical reservoirs. Yeah, they provide a stable structural backdrop that helps maintain predictable neurotransmitter levels and activity in those specific spots. The material even hints areas like the substantia nigra, crucial for dopamine, might rely on these stable patterns.
So it's a way the brain creates pockets of order and predictability within all the constant buzz and activity.
That's the idea. It's how structure might emerge from dynamics.
Speaking of dynamics, the material talks about the brain as a self-organizing system. Lots of high-frequency jiggling, introducing micro-chaos.
Right. At the tiniest scales, things are fluctuating, a bit unpredictable, like static noise.
But it doesn't stay chaotic.
No. The system starts to organize itself. Those chaotic physical wiggles, influenced by the local chemistry and electricity, start coalescing into more structured strain waveforms at a slightly bigger scale. That creates mesoscopic stability. Regional patterns emerge.
And these regional patterns then link up.
Exactly. They contribute to the large-scale synchrony, the big brain rhythms we see on an EEG. That large-scale oscillatory coherence. It integrates the small-scale chaos into the functional brain states we actually experience.
Like being awake or asleep. Which brings us neatly to the sleep-wake cycle. This model seems to offer a new way to think about that transition.
It really does. The material maps out how this physical strain perspective aligns quite nicely with the different states.
So, wakefulness.
Characterized by high dynamics neural strain flow. The brain's physical structure is active, shifting, processing. Chemically, it's dominated by the arousal neurotransmitters, dopamine, acetylcholine, noradrenaline. It's mechanically and chemically geared up for action.
Then we fall asleep into NREM sleep.
And this stage is characterized by strain desorption. The brain seems to be physically releasing or unwinding the tension built up during wakefulness.
Like letting go of stress.
Kind of, yeah. And chemically, this aligns with the dominance of calming transmitters like GABA and serotonin.
And finally, REM sleep, the dreaming stage.
The material describes this as involving reorganized strain flow. The physical patterns are dynamic again, but different from wakefulness, still active. And chemically, you see that surge in acetylcholine, which fits with the vivid internal experiences, even though our muscles are inhibited.
This is the perfect lead-in to your specific questions about dreams. You had this really cool analogy. Fragmental static dreams are like nouns, and episodic dynamic dreams are like verbs.
It's a great analogy, and the material seems to support that kind of distinction.
So fragmental dreams.
Yeah.
These are the static images, right? Objects, maybe just a single scene.
Exactly. Like a noun, they just represent a thing, a state. You might see a locked door or a strange symbol or someone's face just hanging there.
Meaning comes from association, memory triggers. It's like a snapshot.
Right. Reflecting a mental state, maybe an emotion, but it's not moving, not unfolding. It's potentiality, like a noun.
And episodic dreams are the verbs. They have action, plot.
Yeah, things happen. Events unfold. There's progression, interaction. You're running, climbing, talking. Things are changing.
Conveying meaning through the sequence. Cause and effect, maybe, like a story the mind is telling itself, processing experiences.
That seems to be the idea. It's the narrative function, like a verb driving the action.
And you extended the analogy. The mood, the colors, the visual feel, those are like adjectives.
Modifying the noun, the static image, yeah, or the overall scene.
And the pacing of the dream, how you move between scenes, the speed, Like adverbs?
Modifying the verb, the action, how you're running, how things transition. It's a really rich analogy.
Okay, so how does this noun-verb, static dynamic dream difference connect back to the brain kinematics model?
Well, static dreams, the nouns, seem to arise from what the material calls vertical association, linking memory bits based on shared features or concepts, kind of all at once.
Like pulling up related files.
Sort of. And this apparently needs minimal neural strain flow, or maybe just that phase-lock stable flow linked to those local resonances or stationary waves we talked about. It's like activating a single stable memory snapshot.
But dynamic dreams, the verbs, need connections over time.
Right. That requires longitudinal integration, connecting memory units in sequence to build a narrative. And this, the model suggests, depends on active neural strain flow. That physical flow is needed to carry the information and link up sensory bits, emotions, motor commands into a coherent simulation.
Which directly answers your follow-up question, doesn't it? Is the difference between dream types related to the speed, the kinetics of that strain flow? Is it slower in NREM, faster in REM?
Exactly. And the material strongly suggests, yes, that's a key factor. The speed of the strain flow seems to regulate how memory units get woven together. It's like the speed of the assembly line for dreams.
So let's trace that speed across the sleep stages based on this model.
Okay. In earlier deep NREM sleep, strain flow kinetics are thought to be very low or just low. Meaning the flow is too slow or weak to effectively link memories over time. So you get those fragmented dreams, static images, isolated scenes, maybe weird jumps because the connections aren't smooth.
Then later in NREM,
Kinetics pick up a bit, maybe moderate. You might get short, choppy episodic bits, but still disconnected. Like glimpses of action, but no real story.
but then comes REM sleep.
High kinetics. Fast, flexible strain flow. This allows for those fully episodic, dynamic dreams. Smooth transitions, interactive stories, stronger emotions. The fluidity comes from that high-speed strain flow integrating memories effectively along those longitudinal pathways.
And lucid dreaming, where you know you're dreaming.
The idea is maybe very high kinetics, allowing for faster associations, more cognitive control within the dream state.
So it really seems like dream coherence, how continuous it feels, is directly tied to how fast that neural strain flow can move things along and link them up.
That's the core proposition, yeah. The mechanics underpin the narrative structure.
Which brings us to the really deep question you asked. What's actually causing the strain flow speed to change like that? What drives the whole sleep cycle rhythm? Are there fundamental underlying media?
And the material suggests, yes, it's not just random fluctuations. There are likely several interacting fundamental media orchestrating these shifts, underlying the chemical and electrical changes we normally focus on.
Okay, so what are the candidates for these underlying drivers?
Well, one is electromagnetic fields, EM fields, and neural phase synchronization.
The brain's electrical rhythms.
Right. Changes in how synchronized the brain's electrical activity is and the overall speed of oscillations, like shifting from fast beta-gamma in wake down to slower the A-delta in sleep, that likely influences how well different areas communicate and how strain flows can organize. Slower rhythms might mean slower, less coherent strain flow.
Makes sense. What else?
Then there's the idea central to this model, neural strain flow itself and the buildup of residual strain during wakefulness.
The physical debt idea.
Exactly. Just being awake imposes these tiny residual strains. Maybe they accumulate to a point where the system says, okay, need to reset, need to sleep. And significantly, the model suggests the release, the desorption of this strain happens sequentially across the cortex.
Sequentially.
Yeah, starting in the back, like the parietal sensory areas, then moving forward towards the prefrontal cortex as sleep progresses. That pattern of physical unwinding matches sleep stages really well, suggesting strain itself could be a primary driver.
Wow. Okay, what other media?
Cerebrospinal fluid, CSF, and the lymphatic system. We know CSF flow dramatically increases during sleep.
The brain's cleaning crew.
Pretty much. Flushing out metabolic waste like adenosine, which causes sleepiness, and maybe even things like beta-amyloid. Clearing that waste definitely influences neuron excitability and helps lock in the sleep state.
And ions, changes in ion balance.
Yep, ionic gradient shifts. The balance of sodium, potassium, calcium inside and outside neurons changes. For instance, potassium levels outside neurons tend to rise during sleep, which makes neurons less likely to fire, hyperpolarizes them. It's like a global biochemical switch helping to quiet things down.
And of course, the classic brainstem areas.
Absolutely. Brainstem regulatory networks like the RAS, the reticular activating system, and the SCN, the circadian clock. These nuclei are like the master conductors, orchestrating the whole show by shifting neuromodulator balances age-free, noradrenaline, serotonin, and responding to the daily light cycle.
So it's not just one thing driving sleep, it's a bunch of interacting systems.
Precisely, multiple overlapping fundamental processes. But importantly, the model you brought forward really emphasizes how neural strain, its accumulation and release, could be maybe the primary medium linking the mechanical state to the brain's energy use and metabolic cleanup.
Okay, so understanding these underlying drivers helps connect the dots. How the familiar neuroscience story, neurotransmitter shifts, brainwave changes, aligns with this newer strain stimuli gradient to produce the sleep pattern we see.
Exactly. Neuroscience describes the gradient. Wake is high noradrenaline, high acetylcholine. NREM sees GABA and serotonin rise. REM sees A should surge back. Electrically, we go from fast beta gamma down to slow delta, then back to theta beta in REM. Metabolically, high demand, then recovery, then reactivation. Synaptically, strengthening, then weakening or downscaling, then reactivation. It's a whole cascade.
And the strain model mirrors this cascade.
It seems to align remarkably well. Wakefulness, fast strain flows, accumulating residual strain, NREM reduced strain flow, and that progressive release of the stored strain, starting from the back, parietal, and moving forward, a physical relaxation sequence.
And the transition to REM.
Some strain flows start picking up again, maybe a partial reactivation of stored strain patterns. Areas like the prefrontal cortex slowly come back online,
Leading into REM itself.
which involves high strain flow again. But it's described as a reorganization of those residual patterns, not just random activity, but restructuring. This aligns with reactivating associative and emotional networks for dreaming.
So pulling it all together, sleep isn't just rest. It's fundamentally about stabilizing the brain's physical structure, releasing strain, and functionally reorganizing things.
That's the integrated view, yes.
Oh.
The chemical, electrical, metabolic gradients seem to regulate how that physical strain gets distributed and released, mirroring that progressive wave of relief across the cortex. It's a mechanically grounded recovery process.
Let's circle back then to your initial hypothesis, which really sparked this whole discussion. Does the intensity of strain in specific brain locations influence the neurotransmitters there? The type, how much is made, how much is released?
And based on this material and what's emerging in mechanoneuroscience generally, the answer seems to be a pretty solid yes. Your hunch looks well supported. Strain intensity appears to be a significant local regulator of neurochemistry.
Okay, how does intensity affect the release?
Well, think about those mechanosensitive channels again. More intense strain might mean those calcium gates open more often, or stay open longer.
Leading to more calcium influx.
Right, and therefore potentially more neurotransmitter release. Also, astrocytes getting squeezed harder might change how they process and release glutamate or GABA.
Yeah.
And higher strain attracting more blood flow via neurovascular coupling means more raw materials delivered right where they're needed for release.
What about actually making the neurotransmitters synthesis?
Well,
Can intensity influence that?
that's a really fascinating part. The material suggests, yeah, maybe.
Right.
Physical stress can actually activate genes.
Seriously, strain can flip genetic switches.
It seems possible. Strain could potentially activate pathways that upregulate the enzymes needed to make specific neurotransmitters. Imagine intense focus putting strain on your prefrontal cortex. Maybe that boosts the enzymes making dopamine right there where you need it.
Wow.
It could also trigger neuropeptide production in deeper brain regions. And again, astrocytes under different strain levels might change how they take up precursors like choline or tryptophan, or how they convert glutamate into GABA.
So strain isn't just opening the release valve, it might be affecting the factory floor too.
That's the implication. And it goes even further. The material hints strain intensity might even nudge the type of neurotransmitter favored locally.
How so?
The idea is high strain intensity might favor excitatory neurotransmitters, think glutamate, dopamine, ASASH, noradrenaline, the go signals associated with high activity.
And low strain.
Low strain intensity might favor inhibitory ones, GABA, serotonin, endorphins, the calm down or recover signals.
That's a huge concept. Strain could potentially sculpt the local chemical environment, pushing it towards excitation or inhibition based on the physical state.
It could. Think about the prefrontal cortex under high cognitive load. Maybe high strain there favors dopamine and glutamate for focus. Then during rest, as strain reduces, GABA and serotonin might become more dominant for stabilization.
And this loops perfectly back to sleep. High strain in wake promotes arousal chemicals. Low strain absorption in NREM promotes inhibitory ones. Reorganized strain in REM brings back ASHAC for internal processing.
Exactly. Your initial hypothesis connecting strain intensity to neurochemistry seems to fit really well within this integrated framework.
Which brings us right back around, reaffirming that final idea you had this fundamental bidirectional interplay between the physical strain flow and the neurochemical electrical world.
Yes, the material hammers this home. It's not A causes B, it's A influences B and B influences A constantly. A dynamic feedback loop is the core principle.
The physical strain shapes the chemistry release, maybe synthesis, maybe even type preference locally.
And the chemistry, through its electrical effects, influences the physical strain patterns, how they spread, how they synchronize, maybe helping to form those stable zones.
Right, those stationary stress straining waves, they seem crucial as the stable anchors in this dynamic system.
They act as the local physical structures that allow for consistent chemical reservoirs. So a specific area, like the substantia nigra for dopamine, can maintain its chemical specialty even while the rest of the brain is doing other things.
It explains how you get this amazing regularity, this order, emerging from what seems like chaos at the micro level.
Precisely. You have micro-chaos, the high-frequency fluctuations. But then self-organization kicks in at the mesoscale, creating stable patterns, chemical pools, and the large-scale rhythms integrate all of that into predictable states, like sleep cycles. Macro-scale order for mesoscale stability built on microscale dynamics.
It paints a picture of the brain as this incredibly sophisticated, self-organizing system, driven by this constant mechanical, chemical, electrical dialogue.
And understanding this has huge implications, doesn't it? Not just for normal function like sleep and thinking, but potentially for when things go wrong. What if neurological disorders involve disruptions in these stable strain patterns or breakdowns in the feedback loops, leading to chemical imbalances?
Yeah. Here's where it gets really interesting. This isn't just abstract theory. It points towards potentially new ways to think about brain health, maybe even treatments down the line.
Absolutely.
So what does all this mean for you, listening in?
Well, it means the incredible material you brought to us offers this powerful new perspective. Brain function isn't just chemistry or just electricity. It's deeply fundamentally intertwined with the physical, mechanical forces acting within the brain tissue itself.
Right. It means your curiosity about strain led us down this path, exploring a model where the brain is constantly self-organizing. Strain, neurotransmitters, electricity, all influencing each other in this dynamic feedback loop, creating everything from being awake to how your dreams are structured.
And here's a final thought, something provocative for you to mull over, building directly on this deep dive. If sleep fundamentally involves recovering from and reorganizing the neural strain we accumulate, how might actively managing the different kinds of strain in our daily lives impact our sleep?
You mean like physical strain from posture, mental strain from work, maybe even information overload strain?
Exactly. Could managing those inputs consciously, thinking about mechanical brain health, actually improve our sleep quality, our cognitive function? What practical things might emerge if we start thinking about our brain's physical well-being in this way?
It definitely gives you a lot to think about, a whole new angle. Thank you so much for sharing the sources and the sharp questions that made this deep dive possible.
It was genuinely fascinating to explore, really insightful.